Hello and welcome to this session. My name is Katharina Glomb. I'm a postdoc in Peter Ritter's group at the Berlin Institute of Health at the Charité. And today I would like to speak about my more or less recent work on functional harmonics. So let me just share my screen with you. Okay. So my talk is entitled Exploring Cortical Organization with Functional Harmonics. And let me start by telling you what functional harmonics actually are. And just to make it clear, all this work that I'm showing you here was done together with Selene Atasoy, who was a postdoc in Gustavo Deco's group at the same time that I was still a PhD student there. And she turned me on to all this fascinating stuff. So first of all, functional harmonics, very briefly, can be imagined as the equivalent of sine and cosine waves on a brain graph, let's say. So on the left, you can see here an example, a spatial example of sine and cosine waves, in this case of a rope that is being excited by making it swing, basically, with a certain frequency. And apart from the temporal frequency, you can then also see these spatial patterns that the rope will take on, where the parts that are marked with an N will not move up and down, and the parts that are moved with an A are moving up and down, and this spatial pattern is more or less stable. And you can do a very similar thing with a graph, which is depicted here on the right side for a functional connectome. So each dot here in this graph is a functional brain region in the Glasser parcellation in this case, and the connections between them are taken from functional connectivity. So fMRI, bold, resting state, functional connectivity in this case. And Selene and I write about this at length in our paper, which was published last year. And I'm just going to show you some results from this paper and some stuff that we didn't get to talk about in the paper so much, and which I think are the most interesting. So on the brain graph, the equivalents of these spatial standing wave patterns on this very simple one-dimensional rope domain are these kinds of networks, let's say. And there's a number of them, just like you have different frequencies. When you have the sine and cosine waves, you have different frequencies, and I'm going to explain later a bit more what these frequencies are. And you have different spatial patterns that you can project back onto the cortex. And here are just a few examples. There are more of these, and I'm going to get to this later. And this is exactly what we call functional harmonics. Functional just because they're derived from functional connectivity, and harmonics because they are derived from these simple harmonic motions in physics. Just in practical terms, what functional harmonics are, how you can calculate them. So we started with resting state fMRI data from the Human Connectome project. These are a lot of subjects, 812 subjects, resting state sessions recorded for each subject. And then we took the entire surface mesh. So we're talking about a very high spatial resolution here, 60,000 vertices, a bit less. And we used the average dense functional connectivity that is provided by the Human Connectome project, free for download. So we start with this functional connectivity matrix, which is just correlations between brain regions. And we use these correlation values for each of the vertices to determine which other vertices are the 300 nearest neighbors for each vertex. So which have the biggest correlation and which are the most relevant for each of these vertices. So we turned this into a binary matrix. We end up with a sparse binary functional connectivity. And then you compute something that has a very fancy name. It's called graph Laplacian, but it's actually calculated very simply. So you just take this A, this binary adjacency matrix, and you subtract it from this D, which is just a diagonal matrix, which is to say it's zero everywhere except on the diagonal. And in the diagonal, you have the number of nearest neighbors. And it's not exactly 300 each time because well, all this neighborhood stuff is a bit more complicated than it seems at first sight. So it's not fully symmetric, but yeah, the number of nearest neighbors, which is around 300, is in this matrix D. So then you have your graph Laplacian, you perform an eigendecomposition, so something that is also very common, very fast, very easily done. And you end up with eigenvectors, and these eigenvectors are the functional harmonics. Here are a few more of these functional harmonics, again, plotted on the cortical surface. And you have n of these vertices, so about 60,000. And because it's an eigenvector decomposition, you have n eigenvectors. So you also have n of these functional harmonic modes. And of course, not all of them are going to be relevant. So here we're just showing the first 11, and this is also what we analyzed in this paper. But you can go further up, you can use more of them. We computed the first 100, and you can basically go into depth, and it's not exactly clear yet which of them are relevant or how to even really find out which ones are relevant. However, I still think that analyzing and trying to understand the things that the first few of these functional harmonics can tell us about cortical functional organization is very interesting. Okay, so what can we do with these functional harmonics? So first of all, to clarify a bit how to interpret these patterns that I've shown you, that by now you probably think, okay, they look pretty, but what are they? Here on top of the cortical surface, we plot the boundaries of the brain regions that fall into these strong colors at the end of these spectra, just to show you a bit more where we have the visual network and where you have the somatosensory motor and auditory networks, just for illustration. But so what's important here is that these functional harmonics are, in the first place, just a low dimensional embedding of the FC. So they are smooth patterns on the cortical surface, as you can see. And all you have to know is that if two vertices or if two brain regions have same or similar colors, then that means that their functional connectivity pattern overall is similar. So in this way, we kind of extract functional networks by analyzing how similar brain regions are in terms of their overall functional connectivity. So when you look at the first two functional harmonics that are shown here, you find a very large scale gradient. So you have these big networks that all have similar colors, and anyone who knows anything about the functional organization of the cortex will immediately recognize these two big systems, visual and somatosensory motor networks, which are separated in this first functional harmonic and which are then similarly colored in the second functional harmonic and are kind of in opposition to like the rest of the cortex, let's say. And in these two first functional harmonics, we simply reproduced exactly what Margulis and others found in this wonderful 2016 paper. They're switched, but if you look closer, you will see that they are exactly the same. And as they explain and as they analyzed in detail in this paper, you have, if you plot these two functional harmonics against each other in two-dimensional space, so this is this embedding space that I mentioned, you can see the separation between the visual system in one corner, the somatosensory motor and auditory system in another corner, and then higher areas, namely in particular the default mode network in the other corner of this very nice triangle. Okay, so these are the first two harmonics. We reproduced something that is known and that has been shown to be really interesting and relevant and fascinating. But as I said, there are many more eigenvectors and functional harmonics. So we looked at higher ones. And the first thing I think that is very important to understand here is that they are ordered, just like eigenvectors are always ordered by their eigenvalues. And here these eigenvalues are related to the frequency. So just like in sine and cosine waves, we are talking about a kind of frequency, but be careful because it's not a temporal frequency, of course. So we're not talking about oscillations in time, but we're talking about a spatial frequency on the graph. And it's not so easy to see because this graph is, of course, not regular, like it's not regularly spread over the surface, but it's this functional graph that I showed in the beginning. But you can still imagine this equivalence to the swinging rope, which is also a shape in space, right? So they are ordered by increasing frequency. And what does this mean? I think this is a very nice example for this ordering. Of course, here I'm picking one of my favorite examples where you can see this hierarchical organization of brain networks. So we picked 3 functional harmonics that have something to do with the somatosensory motor network. So here in the second one, which I already mentioned, you basically have the whole somatosensory motor network together. It's all similarly colored. So what this tells you is that this network has a common function and has common connectivity to the rest of the cortex, okay? Then if you go to a higher spatial frequency, you see that this higher frequency means that you have smaller brain regions, like smaller regions of the cortex that are similarly colored. And in particular, here you can see how different somatotopic areas that belong to different parts of the body are separated and are placed at different opposite ends of this connectivity spectrum or connectivity gradient. So in particular here, these orange-yellow regions are the hand regions, so the left and the right hand. And this blue region is the face region. So this tells us that the brain areas that are responsible for hand movement and touch basically have a very different connectivity from the brain areas that are responsible for doing the same things for the face, or I think these are the foot areas. So you have an integration here and you have segregation here, but still you see that the hands are similarly connected. If you go to an even higher functional harmonic, you see that left and right hand are differently connected. They have different functional connectivity patterns, which makes total sense because left and right hand can also act relatively independent from each other. So what this shows you is one of the strengths of using functional harmonics or harmonic modes in general, which is that you don't decompose the brain graph into disjoint networks, but you have overlapping networks that have a hierarchical order going from coarse to more finely grained. And one example for this, nice example that I just picked because there are so many nice examples amongst the first 11 functional harmonics that we analyzed, is area MT or V5, which is responsible for perceiving motion. So this arrow just points at this region, and the first row shows you the seed correlation of this region, and you just see this big network that basically extends over big parts of the cortex. But if you now look at different functional harmonics, you see that on one frequency, this MT is very similar to auditory regions, and it also includes this region temporoparietal junction, which is known to be involved in audiovisual integration. So this is basically a network responsible for audiovisual perception. In this 6th functional harmonic, you see the sensory motor pathway. So you see this nice pathway going up to parietal cortex. And in the 8th functional harmonic, you can see the ventral stream, which includes this fusiform face complex, which is responsible for perceiving faces. And interestingly, it also includes the somatotopic face area. So you see how this one region, MT, has different functions depending on which regions you look at. It's involved in different overlapping networks. Okay, the last point that I want to get to is you can use functional harmonic or harmonic modes in general as basis functions. I've already mentioned this, alluding to sine and cosine waves, which you are very familiar with from Fourier transform, of course. But to understand this, I just want to mention briefly that there's a distinction between the underlying graph, which is where the eigenvectors come from, where the harmonic modes, functional or otherwise, come from. And this graph doesn't change, so the basis functions don't change, just like we always use the same sine and cosine waves. but then the signal on the graph, so let's say different activity levels of the nodes of the graph, which signify the different vertices of the functional connectivity matrix, the activity pattern on this graph can change, and it changes over time, or for example, if you look at different task activation patterns. And this is what we did. We decomposed this graph signal that plays out on the non-changing graph into basis functions, just like you do with the 1D signal. And by doing this, you can characterize activity patterns on the cortex by this harmonic modes spectrum. Just like with Fourier transform, you get out a spectrum. And we did this for 47 task maps from 7 different task categories, again, from the Human Connectome project. And we used only the first 11 just to get a nice characterization. So on the left, you can see the original average activity pattern. This is the spectrum. And here we're just showing the composed pattern. So using functional harmonics. And you can see this, I just want you to see here that using the different task categories, you see that these spectra look very different. So you can really say that these functional harmonics characterize different tasks by contributing to them to a different degree to the activation patterns. Okay, as a summary, functional harmonics are eigenfunctions of the resting state functional connectivity brane graph. They embed the functional connectivity into one dimension. Each one of them is one dimension over a range of spatial frequencies, and then you can, of course, combine them. They reflect several organizational features of functional connectivity. I showed you hierarchical networks. I showed you global connectivity gradients. I showed you integration and segregation. And they enable us to understand activity patterns as a superposition of functional harmonics. Open questions, I would say, of course, there are many, not least of all, there are still open methodological questions. So like, what's the best way to build the graph? What's the best way to compute the Laplacian? What's the best way to set parameters that I haven't talked about at all? But more conceptual questions, I think, is stuff like whether we can use functional harmonics to explore differences in network organization between individuals, groups, like patients, for example, or different brain states. And work on this has, of course, already began. I recommend this paper by Selene Atasoy and others already from 2017, where they looked at LSD, which is, of course, a big reorganization of cortical function. So there they showed that you can use harmonics to do that. And something that I'm really interested in is whether there's connection between functional harmonics, the spatial patterns, and oscillation frequencies, like with standing waves, like with the ropes that I showed you in the beginning. And also here, work has already begun, and there's this modeling paper from the Raj Lab from 2020. And I recommend looking into that if you find this topic interesting, like I do. And to end, I just want to thank the co-authors involved in this paper that I've mostly talked about today, and I'm open for your questions. Thank you.